Now, I'd like to invite Jim Goss, who is the scientific director of the foundation, to give a talk on introducing biomarkers. Thank you, Jim.
You're welcome. So it's kind of nice I can stand up here and now push this button. I've been coming up here for the last 2 days because we've had our scientific conference the previous 2 days that I would come up here and introduce people and then watch them be able to present. And so now it's my turn. And so what I'm going to talk about, we're actually going to have 3 talks on biomarkers. And so my talk is I'm going to kind of introduce them and tell you why they're so important and how you are helping us in this and how you can continue to help us in understanding and identifying biomarkers. And so basically just an outline of my talk, I'm going to give an overview of what biomarkers are, then I'm going to talk about potential biomarkers, not in too much detail, and give you an example of one biomarker or one study that we've done using blood that we've taken from you and have utilized. So what is a biomarker? So a biomarker just means it's a biological marker. It is a characteristic that can be objectively measured, which means that it's something that you can actually put a number to if you want to think of it that way. It's evaluated as an indicator of either normal biological process or of a diseased biological process. And it can also be utilized to understand if a drug is having an effect. And so an example of a biomarker that everybody is probably very familiar with is blood pressure. Everybody's probably had their blood pressure taken. That's a biomarker. It's objectively measured. It gives you numbers and it gives you some idea of whether or not your cardiovascular system is working correctly. Cholesterol is another one. So every time you go have blood work at the doctor, they're basically collecting a lot of values which act as biomarkers to tell the physician how healthy you are if you're having any problems. And biomarkers can be molecular, like cholesterol. Biomarkers can be a structure, like you can take X-rays or MRIs. That kind of imaging can be utilized as a biomarker, or they can be a process, and like blood pressure is a process in the body that can be evaluated. It's important to say biomarkers are not symptoms, so they're not things on how you feel. They are things that are objectively measured. So the FDA recognizes 7 different classifications of biomarkers, and you can see them on the left side of the image here. Those classifications range from biomarkers that basically can test whether something is safe or if you've been exposed to something that's toxic, and something like that might be lead in your blood if you've been exposed to a lead-based paint. They can be diagnostic, which means it's a biomarker that shows if you have some sort of disease. They can tell you there are susceptibility-based biomarkers, which can be utilized to say if you are susceptible of potentially getting a particular disease. There are some cancer biomarkers that might suggest that you might be more susceptible to getting a type of cancer, for example. There are prognostic and predictive biomarkers, and there are biomarkers that we use to see if somebody is responding to a medication or to monitor either how they're responding or how their disease is either stable or progressing or getting better. As I said, there can be many types of biomarkers, but to have the kind of biomarkers which are the most useful, you want to look for some particular types of characteristics. And so one of those is that the expression is significantly different in the biomarker if you're going to be using it to indicate a disease. It's got to be different in the disease, if somebody has a disease, than it would be otherwise. Another good characteristic is it's got to be a biomarker that you can readily quantify, and and you have to quantify it for something that you can readily access, right? And so, like blood. Blood is easily, it might be a little painful, but you can collect blood from almost everybody, and so you can look at biomarkers in blood. So those are very good biomarkers. You know, CSF is more difficult, but it's not a horrible biomarker, but it is more difficult to obtain, right? But you're not going to get a biomarker where you're going to drill a burr hole into your brain and collect tissue. That's not a good biomarker. That's not accessible, and so that's not going to be usable. You also want biomarkers that are going to be economical as well, and ideally, if you're trying to monitor how a disease is progressing, you want a biomarker that reflects what are known as clinical outcome measures. Ways of demonstrating that, in fact, the disease itself and the symptoms are improving in a disease. So biomarkers are used in three main ways as far as we're concerned. So one is disease management. And again, this is diagnosing the disease, predicting the disease, how somebody is either recovering from a disease or a disease is progressing. Another is treatment efficacy. This is probably for us the most useful biomarkers that we're going to see in the coming years or for STXBP1, and that is basically, is a potential therapy working, early detection of efficacy in a therapy, optimizing treatment for a therapy. And then biomarkers can also be used in clinical trials. So for some clinical trials, instead of actually demonstrating that the person is, instead of showing that you're having an effect on a person's symptoms, if you can show that a biomarker is changing, that's good enough for the FDA. Okay, those are very difficult biomarkers. We probably won't be able to have something like that for STXBP1, at least for a while. But importantly, kind of that second set with the treatment efficacy is probably the types of biomarkers that will be most useful. Why do we need biomarkers for STXBP1? Well, one is that, as you all know, there are a wide range of symptoms in people who have STXBP1, and those symptoms vary, and because those symptoms vary, sometimes it's difficult to just look at symptoms to see if somebody if a therapy might be working. And so if we have a biomarker that is in common for all STXers, that makes it much more easy. Another is what's called target engagement. And so all drugs target something, that's how they work. And a biomarker can be utilized to determine if a drug is actually hitting the target. So if your target is to, for example, targeting the gene for STXBP1 to make more STXBP1, then a good biomarker, for example, might actually be, can you measure an increase in STXBP1 in some sort of fluid or part of the body that you can assess? A couple other reasons our biomarkers can be utilized to select patients in clinical trials. Sometimes this is more important in very early clinical trials when you want to try to treat patients who are all fairly similar because you're trying to find effects that are the same. It makes it easier to get a drug approved, and then you can expand the drug use to other individuals. But another important one is dose response time. So STXBP1, as a neurodevelopmental disorder, the issues that we see in the population, if you treat with the drug, it may take quite a while before you start to see advantages in the efficacy of a drug, because you have to start making STXBP1, and that has to go into the nerves, and that has to start increasing neurotransmitter release, and a bunch of things have to happen in the brain before you may actually see really positive effects in the symptoms in our kids. but a biomarker might change very quickly. And so that tells pharmaceutical companies that, yes, their drug is working and we're going to continue the clinical trial and we can be hopeful. There are several types of potential biomarkers. There are biochemical biomarkers, and these are metabolomics, or basically how metabolites that can be measured in your blood, proteins, RNA, transcripts. There are electrophysiological biomarkers like EEG, functional biomarkers like how somebody's eyes may track, for example, or wearables. We'll hear more about this in the next talk. And imaging biomarkers. So how can you help us discover biomarkers? Well, you do it by this. You draw blood, or somebody draws your blood, collects plasmid at the family summit, which means go get your blood taken. We need blood from STXers, but we need blood from controls. Control blood is very important. And I hate to put this on the siblings, but the sibling blood is the best blood. We take parent blood, but sibling blood is great because we really need age-matched controls, or the best controls, and family controls. And I'm being told I have 3 minutes, so I will move on. Anyway, we measure differences, and that tells us. And so I'm going to briefly discuss a study on how we've taken your blood and started to look at that and gotten a potential biomarker. There's more to this, but so we work with Combined Brain. They're the people who are taking your blood, by the way. And in this particular study, they did a study where they were looking at changes in protein. But it wasn't just an STXBP1. It was from 31 different neurodevelopmental disorders that were collected. The blood that they used for STXBP1 was collected during the Family Summit in 2023. And they took the blood, the plasma, and they measured over 10,000 different proteins. And we had, in our STX population, we had 23 affected samples that they took and 7 control samples that they took. And if we just look at STXBP1, there were 10,000 proteins, but I'm only going to focus, I'm not going to talk about all 10,000 because I only have 2 minutes now. But so I'll talk about STXBP1. So you can see that there's a little difference here, right? And we're kind of hoping STXBP1 is a haploinsufficiency. Ideally, what we would like to see is half the amount of STXBP1 between an affected individual, if somebody has STXBP1, and a control individual. We didn't see that. We saw a little bit more in the control. But the thing about biomarkers is you need more samples. So the more samples you have, the greater chance you're going to see if there really is a difference. And so I said that they collected blood from 31 different disorders, and all of those disorders were not at our family summit, in case you weren't here in 2023. It was really just us. So what happened is they went to a lot of different family summits. So all of these neurodevelopmental disorders, have family summits like we do, and they go and they collect blood from them. In fact, they went to 25 different sites and collected blood. And so if you look at the STXBP1 levels from the blood collected at all those different sites, what you'll see here, and I hope you can appreciate this, is they're very different based upon the site. And it turns out what this tells us is how you collect blood appears to be very important for being able to measure STXBP1. And I think Mike, who's going to be talking next, will go explain this maybe a little bit more. But if we look at this, so where I have this circled, this is site 6. This was our summit. So, and that's the STXBP1 levels collected at our summit. Then there's a dotted line, and we had 1 individual collected at that summit. Now I now have 1 minute, but that's okay. But control samples can actually be utilized from any control, right, as long as you don't have STXBP1. And so if we go and we look at the control samples from these sites where the STXBP1 level matches closely the STXBP1 level from the site where it was collected for us, which is site 6, then what you see is on the other side, so now we have the STXBP1 affected from our site, our controls at our site, and then now we have 88 controls added from the other sites that had similar collection properties to our site, and now we see there is a difference in STXBP1 in the plasma between controls and the affected. And just, I won't talk about, I thought I wouldn't talk about 10,000 proteins. I'll briefly state, I have 0 minutes now, I'll briefly state that here are 3 interesting proteins where there was a difference even just within our small sample. So neurotensin, myelin transcription factor, and the lovely name PDZ in limb domain protein 5, which I know everybody understands what that is, but these are all involved in neuronal processes, and especially this last one, which is involved in synaptic structure development. So there's more to do, and we've got a lot still to learn, but we only learn this if we get your blood, and I can guarantee you your blood gets used. I see and I approve every sample of blood and plasma that Combined Brain, who keeps our blood, sends out to pharma and sends out to other researchers, and they send out a lot. And so we need to keep refilling the bank. And so again, please go donate. You know, if you don't have a sibling, go out into the street and grab some teenager and bring them in. So don't do that, really. So.
Is there any question? We have time for one question for Jim. All right, if not, you can grab him during lunch. Thank you. Oh, is there a question? Sorry, I couldn't see.
Yeah, I was curious to know, like, the blood sample that's collected of the patient and the siblings. Do we get to know anything about what you guys do with it and what's the result of it or outcome of it?
So that's an excellent question. And so when you give your blood, basically how it'll be collected is they use what's called a CRID number with Combined Brain. So if you've already given blood, you have a CRID number, you've signed up for that. If you haven't, that's one of the first things that they'll do. That makes your sample anonymous. And so when your sample gets put into the biorepository, and that's what we call where we put all of the blood samples, and if you've given other types of biological samples, they all go into this biorepository, and it's only identified by CRID number. And so and the only thing we know about the CRID number is whether or not somebody is affected or a control. We know age, and there are a couple other things, but it's meant to be so that we can't identify. We don't keep the CRID number. The foundation does not keep the CRID number, so that anything that you provide is anonymous. And when we send samples out, we're just sending them out based upon somebody might want 20 affected, 20 controls, age matched. And so we can go in and we can try to match samples to what they want, but we don't know actually who the donor was for those. So.